इस बात का बारहा जिक्र हो चुका कि हम जेल इलेक्ट्रोफोरेसिस करते हैं। जेल इलेक्ट्रोफोरेसिस क्यों चाहिए, क्यों इस प्रोसेस की ज़रूरत होती है? इस प्रोसेस की ज़रूरत हमें है टू स्टडी एनी मैक्रोमोलिक्यूल लाइक डीएनए, आरएनए और प्रोटीन। अभी हमने पीसीआर के हवाले से बात की पिछले दिनों। जेल इलेक्ट्रोफोरेसिस is required to study the DNA sample. Now we are going to see how many types of gels are there, what are the advantages or disadvantages of any gel which can be used to study the DNA. Electrophoresis is heart of the scientific study of DNA fragments. As the name indicates, it involves some electric current, and the purpose or the result of this method is that the different sized DNA molecules are separated under the electric field by this process. This is a simple method, rapid, and is capable of separating the DNA molecules which otherwise could not be separated by density gradient centrifugation. Density gradient centrifugation is a process which separates the molecule on the basis of density. So different DNA molecules may have same densities but different molecular weights, so it is difficult to separate them by that method. Therefore we have to use electrophoresis for this purpose. After doing the electrophoresis, how we can locate the DNA in the gel? For this purpose जेल स्टेनिंग इज डन। और पहले भी बात की थी कि डीएनए इंटरकेलेट्स द एथिडियम ब्रोमाइड, और जब इस एथिडियम ब्रोमाइड की मौजूदगी में डीएनए को एक्सपोज किया जाता है यूवी लाइट के साथ, इट गिव्स यू शाइन, फ्लोरिसेंस. सो स्टेनिंग विद एथिडियम ब्रोमाइड makes it possible that we can identify the presence of DNA in any gel, and by this method we can detect as minimum possible 20 picogram of DNA in any gel. Two types of gels are used. One is agarose gel. The second one is polyacrylamide gel. The gels, they vary in shape, they vary in size, and depending upon the percentage of the gel which we made, हम इसके ऊपर बात करेंगे, depending upon the concentration, the pore size of the gel differs. It depends on the pore size that allows what molecular weight will pass through that gel. अगर हम agarose gel की बात करें, this has the lower resolving power. It's not very good as far as resolution is concerned. However, it has a greater range. हम इस gel को use करते हुए 50 base pair से लेके several mega bases तक का जो DNA है उसको separate कर सकते हैं, depending on the size, concentration and configuration. अब configuration की जब बात आती है DNA में, एक आपके पास linear DNA होता है, एक plasmid DNA है which is circular DNA, फिर supercoiled DNA आ जाता है. So keeping in view the configuration of the DNA, the percentage of the gel is decided. By agarose gel electrophoresis, we can separate the DNA, and this gel is run horizontally, whereas the other gel, the polyacrylamide gel, is run vertically. For this purpose, here we see the gel apparatus, the apparatus for gel electrophoresis. This blue and red comb is going to be used to make the wells during the gel casting. हम इसके बारे में बात करेंगे. These are the gel trays which will be used for again gel casting, and this chamber is going to be used for electrophoresis. This red color and black color are those terminals which will be connected to the power supply to provide the electricity, and inside if you see this red color and blue color, these are again the combs which shows you the presence of the gel casted in the tray. Polyacrylamide gels, they are more effective in separating smaller DNA molecules from 5 to 500 base pairs. They have extremely high resolution or resolving power. and they separate the DNAs even which differ in one base pair. These polyacrylamide gels, they are run vertically. Like here, the sample is being loaded and the direction of electrophoresis will be from top to bottom. There are advantages and disadvantages of one type of gel over the other type of the gel. For example, this gel runs very quickly, the polyacrylamide gel. It accommodates comparatively larger quantities of DNA. It runs vertically. However, this is more difficult to prepare as compared to the agarose gel. This figure shows you all the requirements for casting the gel, the polyacrylamide gel, and for doing the electrophoresis. This is the gel casting holder. The green one is the glass plate holder. These glass plates are basically used to cast the gels. These are the combs, and these colorful holders are basically the guides when we are going to load the samples. This is the tank in which the electrophoresis is done. Gel composition plays important role. We have to be aware of the size of the DNA molecule we are looking forward or we are studying. Depending upon the size, we determine the gel. As the molecular weight of the DNA goes up, the percentage of the gel should go down, because when you decrease the percentage, the polymerization of the gel is going to give you larger pore size through which the larger molecular weight DNA can easily pass through. These low percentage agaroses, they are fragile and long times are required to do the electrophoresis, sometimes several days, but there is size limitation which the DNA can be separated in these gels up to 750 kilobases, but sometimes the process also fails. The electrophoresis fails to give you separation of the DNA molecules. Keeping in view this problem, for larger DNA molecules, the scientists Schwartz and Cantor in 1984, they developed another electrophoretic method, which is pulse field gel. In the pulse field method, we use two sets of electrodes, and in this case they are at diagonal position like this. The flow of current alternates. For some time it alternates between these two pairs. Sometimes it is moving in this direction. After some time it starts moving in this direction, and it can make it possible that the large size DNA fragments can be separated on these gels.